Okay, so let's look at multiple alleles in detail. Okay, so a multiple allele is you already know it's a alternative form of a gene. Okay, or a contrasting form of a gene, or a alternative form of a gene, or a copy of a gene is an allele. Okay, so here you can look in this picture. You can see that this one, it means one chromosome. This is another chromosome. Similarly, this is one chromosome, another chromosome, one chromosome, another chromosome. And these are homologous pair. These chromosomes are homologous, these chromosomes are homologous. Now, this one at this place, what do we call this? Remember? Locus. Okay. So, at that locus, you have in one chromosome this plus allele, and another one is another plus allele. So, this is a homozygous gene. Okay, this is a homozygous. It's in homozygous pair and the wings appear like that. Okay, and another form of a gene, another allele, which also codes for wings. But the name is VG. Okay, VG. This first allele is coded by plus. And here second is coded by VG, third is coded by VGA and just blank. So now here you can see very clearly, that here this gene, all the three genes, okay, all these three pairs of genes, they code for wing shape. But how the wing will look, okay, whether it's long or thin or feathery, okay, it is not the job of the gene anymore. The gene says you will code for wings, okay. But because of the alleles, how the alleles are arranged, if in one homologous chromosome is plus and in another also is plus, the wings appear normal. And in another one, if the gene, okay, if the gene is of VG, the wings look small. And then another one, this one VGA and plain, the wings look, I don't know, is it broken wings? I don't know, maybe. Okay, so just from that image, it shows you that how the multiple alleles, how they express, okay. I think it's already clear from remember when I explained about the flower color? Okay. So there are 3 or more kinds of genes occupying the same locus. So here the gene for a wing shape, it is coded not just by like tall for T. We study like this, tall for T and small t for dwarf. But here the wings, the genes could be, we saw what? Plus, we saw VG, we saw VG superscript A, and we saw just empty. Okay, so see, there are 4 different types of expressions. So these alleles are 4 in number. Okay, and how they they come together, that is what gives us the type of shape that this Drosophila wings will have. So 3 or more kinds of gene occupying the same locus in an individual chromosome are referred to as multiple alleles. Okay, they should occupy the same location on the chromosome. Okay. In short, many alleles of a single gene are called multiple alleles. Okay. And the concept of studying multiple alleles is known as multiple allelism. So what it means is that one gene can appear in many forms. Okay, like this, or like this, or like this, or like this moon shape. So one gene, it can appear in many forms. Okay, so that is your multiple alleles. Okay, one gene can appear in different forms. We saw plus, we saw VG, we saw VGA and blank. This is nothing but that only. Okay, one gene for what was that? Wing. Gene for wing shape. It could express itself in different ways. So the characteristics of multiple alleles are that we have to study them in the population, and they are situated on homologous chromosomes at the same locus. So there should not be any crossing over between members of multiple alleles, because they have to be in pair, no? They have to pair up. They have to pair though, like this, no? They have to pair up. There should not be any crossing over, okay? If this is one allele, this is one allele, they should not cross over. This is one allele, this is one allele. They will cross over between different alleles, okay? They cannot cross over. And multiple alleles will influence one character only, whether it's like hair color or eye color or shape of flower, shape of wing, okay? Multiple alleles, they will complement each other, and the wild type allele is always the dominant one, okay? So what we find in nature, the wild type is the dominant one, and the other mutated alleles, they are less dominant. They could be dominant, or they could be recessive, or they could be co-dominant. But the wild type, the normal type, the one we find in nature, the one that was groomed by nature, is your dominant one, okay? So when we perform hybridization, when we perform crossing over, we get mutant types when we hybridize something. So these are not wild types, okay? The mutant type, the hybrid ones, these are not the wild type. They're not the ones found in nature. It's because of, human intervention. Okay, so let's look at the examples of the multiple alleles. The first is the wing of the Drosophila we had seen in the image. Well, usually in a normal Drosophila, the wings should be long, okay? Then some mutation occurred at the same locus in different flies, okay? One got reduced wings, the VG, the vestigial wings, and the other got antlered wings. Remember those broken looking wings? So both the vestigial and the antlered are alleles of the same normal gene, and also for each other, and are recessive to the normal gene, okay? So if there was a plus sign and a vestigial one, this dominant will express. So it will be a normal wing, okay? But since they were in the homozygous state, then the vestigial, that small wing became, and that vestigial and blank, it showed that antlered one, the broken wing, okay? Okay, so here they are showing you the three races of Drosophila. The long wings have a plus homozygous alleles. The vestigial have a VG. The antlered have a VG antlered, okay? So a cross between a normal and a vestigial or antlered is shown below. Okay, so this parent, one parent has a normal and one parent has a vestigial wing. So on selfing, we will get, the F1 will get long wing, because one is the dominant one, Then on selfing, after successive crosses, you will get 3 long wings and 1 antlered wings, okay? This is your basic Mendelian genetics, okay? You cross them, and whichever is dominant, you express that. Then see here, you are getting normal long wings. You are getting here again normal, because the dominant allele is there, the plus one, and only here this one, it is the antlered one, okay? So these are the experiments that they performed to show multiple allelism, okay? You will understand how to do crossing better when we do the gene interaction. I will show you how we are doing, okay? I'll show you line by line how to practice it. So when we cross a vestigial wing with an antlered wing, the F1 will have intermediate in between, okay? That will show that none of the mutant genes is dominant over the other. So this is another cross here where they have crossed the vestigial with the antlered, And not only did they show vestigial and antlered as the parents, but they showed one intermediate, okay, which is in between a vestigial and an antlered. Okay, now let's look at the coat color in rabbits. So the skin color of rabbits, they are influenced by so many multiple alleles, because you have seen, no, some rabbits are white, some are brown, some have patches of white and brown, you know, some are gray color, okay. So the normal color of the rabbit, like before, you know, in nature when they used to hunt rabbits, no, hunting used to happen, the normal color should be brown, okay. But there were some albino rabbits, okay. There were some albino rabbits. Those white rabbits, those are the new ones, the mutant ones. There are some albino rabbits. There are, besides it, there are white races called the albino and Himalayan as the mutant race, okay. The Himalayan is similar to the albino, but the ears, the nose, feet and tail are dark, okay. So just think about a white rabbit, but the nose, ears and feet, they are like brown, okay. But the albino is fully white from head, nose, tail, foot, whatever. So the mutant genes of albino and Himalayan occupy the same locus and they are alleles, okay. And both the albino and the Himalayan are recessive to the normal existing one, okay. The one that exists in nature, the plus. So again, you cross the albino and Himalayan and you will get 3 Himalayan and 1 albino, okay. Okay, in the first generation, in the first F1, when they cross the albino with Himalayan, in the first F1 you got the Himalayan. You see there? Okay. So then we can say that Himalayan is more dominant to albino, but obviously the normal one, the plus one, the A plus is the most dominant. A plus is most dominant, the one found in nature, the brown. That is already taken, okay, that we have already accepted, that the one in nature is the most dominant one. But if you cross between these two, a hybrid will appear, which will appear Himalayan, because this one is more dominant to, Himalayan is more dominant to the albino, okay. Then again, if you self them, then you will get 3 Himalayan and 1 albino, because only the recessive genes, when they are inhomologous, then they will show the albino, okay. This is very easy. It's your dihybrid cross, simply a dihybrid cross, okay. Then the self-sterility in plants. So I think you will do with another miss, you will do about self-sterility. Now Kjolter, he describes self-sterility in tobacco, and he described that there are series of alleles that are designated as S1, S1, S2, S3, S4. These all denote for fertility in plants, okay, or sterility in plants. Okay, let me repeat again. There are some alleles, S1, S2, S3, so and so and so on and so forth, that that are the gene that codes for sterility, okay. Sterility in plants, okay. So that part is clear. For self-sterility, now you have a hybrid, okay, S1 and S2, okay. This is not a hybrid. S1 is not a hybrid. This is a homozygous. No, hybrid is the new one. Okay, the S1 and S2, or S3 and S2, or S4 and S1. Okay, it should be mixed. You got it? The hybrid should be mixed up. Then these hybrids are self-sterile because the pollen grain from these varieties, they don't develop. Okay, that pollen tube, they will not germinate. But the pollens of different hybrids could fertilize. Okay, I think I better just draw. You are not. I can feel myself. You know, it's not clear. So let's say this is a pollen S1. Okay, this is a pollen S2. No. hybrid. Remember hybrid, it should be in pairs, no? S1, S2 pollen and S3, S4, this is also another pollen. These are pollens. They landed on the style, okay? This is the style. Then they develop a germ tube. No? And then let's say this gynosium, this one, this vase, this gynosium is S3, S4. You getting? This gynosium has the alleles that is of S3, S4. Now, when the pollen of S3, S4 land, means its own pollens, okay? This is the androecium, okay? This is the androecium. And by chance, this pollen fell on itself. It fell on the style of its own self, okay? Of its own gynosium. So, even if this pollen tube germinate, this ovary and ovule, they will reject the S3, S4 pollen tube. But if this S1, S2, it came flying in the air, no, it landed here on the stigma and it germinated the pollen tube, it will allow the pollen tube from S1, S2 to germinate and fertilize the egg of S3, S4 of itself. You get it? So, similarly, just suppose we have S1, S2, ovule, okay, this one in the female, and then S3 and then its own self pollen came, S1, S2, it will reject, okay, it will not accept, it will not allow the pollen tube to grow, okay. But if another plant's pollen flies in the air and lands here, let's say it's S5, S6. Okay, another plant's pollen lands here, S5, S6, it will germinate and it will fertilize the egg, okay. Fertilize the egg, fertilize the ovule and produce the seed and the fruit, okay. So, I think you have understood the self-sterility in plants, okay. It will reject its own pollens, okay, because nature has designed itself in such a way that it always prefers to get cross-pollinated, okay, so that these dangerous recessive genes do not express, okay. That's why we don't marry our cousins, that's why we marry people outside of our family, no, so that these dangerous lethal genes do not express, okay. Some you have heard, no, some deformities happen when their cousins intermarry, okay. So, same nature, we have to be told, no, by elders about this, by science we have to be told, but nature by itself, it is already designed that I will not accept my own pollens to land on my own style, okay, because cross-fertilization is always better to produce better and stronger hybrids, okay, better and stronger F1 generations or F2 generations or infinity generations. It's always better to cross-pollinate, okay. You get better genes, you get stronger genes from outside. Okay, so let's read what is written here. The genes causing self-sterility in plants probably produce their effects by controlling the growth rate of the pollen tubes. I've already told you that they control the pollen tube, no? The pollen tube grows more and more rapidly as it approaches the ovule, but in the non-suitable one, the growth of the pollen tube will slow down, okay, and it will wither away before fertilization can even take place, okay. So, it will not allow, okay. If it is the right one, if it's the cross-pollinated one from a distant plant, it will allow the pollen tube to germinate, but if it's its own pollen tube, it will not allow. So, now let's look at another example of multiple alleles, the blood group in man, okay. So, all of us, we have one gene that determines blood group, okay, but whether it is which group of blood, is it A or B or O, it is the alleles which determine it, okay. So, we have 4 blood groups with regard to the presence of the antigen A or B or absence of antigens, which could give you the O, or if both are present in the antigens, it will be AB blood group. Okay, so these are how the genotypes are appearing. If it's the genotype is AB, blood groups AB. If it's homozygous or heterozygous for O and A or O and B, it will be B blood group. And if it's in homozygous recessive condition, it will be O blood group. Okay, so here O is the most recessive. Okay, and A and B are equally dominant. Okay, so it appears to have an O blood group if the genotype, here I'll just explain this part of the table. The person will appear, means the phenotype, the person will appear to have O blood group if both the genotypes are in homozygous recessive condition. Okay, and then the person could be an A or a B blood group if it is if one pair, if it has homozygous O or A. Okay. All right, or homozygous B or O. Okay, because O is what to say, recessive. Should I draw or what should I do? I think it's clear. If you have any doubt, you can ask me. Okay, it is very easy. You don't have to draw the full table. You can just show a few. You can just show 1, 2, and 3. You can just show 3 or 4. You know. You can just show a few if you want to see also. If you want to show also, you can just show a few. You don't have to draw the whole table unless you have so much time, no, to write in your exams. Now, I think the AB and O, ABO blood group is clear because you've done it in zoology, I think. You've already done it. But you might not be clear with the rhesus blood group, okay, RH blood grouping. It's more complicated. So I will explain this one better. Okay, if you read this note, no, you'll be super confused. So write the simple one, which I'm going to say. So this rhesus blood group was discovered in the blood of the rhesus monkey. Okay. And it is present in all humans. Okay, so you may have noticed when you did your blood group, you were B positive or A positive or O negative or O positive, you know, so or AB negative. So that negative sign, it shows whether you are RH positive or RH negative, or you are rhesus negative or rhesus positive. Now this allele for rhesus, okay, the RH positive or RH negative, it doesn't have only 2 alleles. We used to think that the allele will be either positive or negative. You know, you are either A positive or B positive negative. No, it's not that there are only 2 alleles. It has many more alleles. In fact, the RH positive, okay, so these two scientists have given their own symbols. So let's look at the simpler one, no, this RZ, okay. Now, if we look at the scientist's Venus symbol, for the RH positive, there are so many sub-alleles. How many? 1, 2, 3, 4. So, there are 4 alleles which express whether the blood group is RH positive. Okay. So, if the person has the blood A and RZ, it will be A positive only, but the allele is RZ. Okay. And if the person has, let's say, AB R double prime, it is R negative only, but it has a subset of the allele of R negative. Okay, so it could be negative, could be R or R double prime or R single prime or RY. Okay, so it doesn't mean that there's just 2 alleles, plus and minus. Okay, we know that alleles are like this, no? They are just 2 alleles. Gene for tall. Okay, it has 2 alleles expressed by capital T and small t. Here, there are many alleles, okay? So Rh positive, not only is it already a multiple allele showing different characters, different traits, but it has different alleles, okay, subset of alleles. Okay, now if the mother has an Rh, okay, let's look there. Okay, now here, what is dangerous is if the person is rhesus negative. Okay, so could be R prime, or just R, or R double prime. Okay, so any of these alleles, whether they express, or R0, I think so, any of these alleles would mean that the person is Rh negative, right? So what is dangerous is if the mother is Rh negative. So you understand, no, the different alleles which could express negativity. So these are the 4 alleles. Okay. So if the mother is Rh negative, and she's pregnant with a baby who is Rh positive, so it could be, remember, RZ or R prime or R2, remember? Okay, so if the mother was Rh negative and the baby inside her tummy is Rh positive, so what happens is that positive and negative, they do not understand that they are from the same. They do not understand, okay? They think that this positive, Rh positive, is a mutant, or is a virus, or is a bacteria. So what happens, these Rh negative antibodies from the mother, they go into the fetus and they attack the blood cells of the infant, of the baby, and they try to destroy all the red blood cells. Okay, because why? In this mother, it never knew about Rh positive. It was always Rh negative. The whole body is Rh negative, so it never experienced this Rh positive, no? So what happened? So in this mother who is Rh negative, so these Rh negative antibodies, they will attack the infant red blood cells. Okay, all these blood cells, they contain the blood group as well as the Rh positive. So what happened is that when these antigens, they attack the red blood cell, they destroy the blood cell. So wouldn't the baby suffer from anemia or other diseases, no? Or hematolysis is when the blood, they explode, right? So it will not be able to develop, because everything, the placenta, it sends blood and nutrients, no? And the baby will need its own blood also to grow. So if these red blood cells are destroyed, that baby will die, right? It will lead to infant death. Okay, but medical science now, it says that you can inject the mother with some serum, okay, with some serum which will go and bind with the Rh negative antibodies so that they cannot attack anymore. Okay. So they have to immunize, just like you keep injecting, no? You have to keep immunizing, you have to keep injecting the mother with anti-sera, okay? And these anti-sera, they come in different symbols, no? All these CDE, big E, small e, which will react with the different kind of antibody that it needs to protect itself, okay? So that the infant will be protected. So if you inject with these different kinds of anti-sera, the developing fetus will be all right, okay? Because all those antibodies who thought it was a foreign, it will be it will be blocked, okay? So this is the most simple way I could explain. I don't know if it is okay with you. You can always YouTube, some better explanation, okay? So there are some importance of multiple allelism. So one is the pseudo alleles. Alleles are different forms of the same gene at this or the same locus. Look at the same locus. Now, pseudo alleles are just like alleles, but not exactly. Let's say these are alleles. This is allele A, and this is allele A. They're at the same locus in the homologous chromosome, but and it codes for, let's say, skin color. Okay, but let's say nearby there's another allele, which is B. Okay. It is nearby, it is not on this exactly on the same locus, but it's very near, okay, just maybe microns different, okay. It also codes for skin. So whether A express or B express, it will still show skin color. So they behave as pseudo alleles. You get it? So if something happens with this A, so this A and this B will combine together and show the skin color, okay. So they are very closely linked to the actual allele. And we can call them as pseudo alleles because they show the same character, okay. Another one is penetrance and expressivity. A recessive gene produces its phenotype only in homozygous condition, and the dominant produces whether it's homozygous or heterozygous, okay. Then sometimes some genes will fail to produce their effect when they should, okay. The ability of the gene to produce its effect is called penetrance. Sometimes if you change the environmental condition, okay, you change the moisture, you change the light, you change the temperature, okay, you change the soil, the gene will express differently in different environment, okay. So example was of this cotton plant which produce a wrinkled leaf, okay. When it's produced in normal season, this cotton plant is grown in the normal season, the leaf becomes crinkled. But if you delay their fruiting, they will not show the crinkled nature, okay. So if it shows that it doesn't penetrate well, that crinkle expression of the gene doesn't penetrate well in the off season, okay. So you can read the other example by yourself, the lima bean example. Let's look at the isoalleles. Well, isoalleles means something which is like your mirror image, no? So a dominant gene can occur in two or more forms, okay. Multiple dominant alleles will produce the same phenotype effect, but the effect will show a small difference in heterozygous state. Okay, the example you can read on your own, and then the phenocopy, xenia and metaxenia, again you can read on your own. Phenocopy is when there is an interaction between the genotype and the environment, okay. When you change the environment, the phenotype expression is different, okay. Metaxenia is when a foreign pollen or a foreign tissue, it falls in the endosperm and the embryo, but even though it's a foreign pollen, it still shows some effect, okay. So the self-sterility, it is something to do almost like self-sterility, okay. It's a foreign pollen, like let's say this is a corn, but a pollen of a orange fell on it, okay. It still can show some effect, okay. Instead of like a yellow corn, you will get a purple, sometimes yellow, sometimes blue, like a mixture of colors. So it is these foreign particles that they showed some phenotype effect. So that is your metaxenia. Xenia is again when you have a foreign pollen, it will show some effects on the endosperm, okay. So I think this is done. This chapter is done. Now I am rushing a lot because we have to finish your syllabus in 2 months. No, you have your exam by June, I think, or July or something. I don't know. But you should start studying, girls, just because, it's just the start. I know it's boring and you're tired, no, to start your studies. But the earlier you start, the better so that you can finish studying for the exam and you can start preparing for your entrances.